Hello everyone, just a small correction, I'm from Portugal. I did my PhD in Barcelona, but that's fine. And I'm working at the Department of Psychiatry in Oxford and also at the Life and Health Research Center in Portugal. So today I'll talk about models of network dynamics. And if you're wondering how this figure is done, it's actually made from real fMRI data, and I'll explain this on Tuesday morning in Andrew's symposium on dynamic functional connectivity. But today I will focus on something else. Okay, so before I start, and since this is the last talk of the day, and maybe some of you are a bit jet-lagged or anything, I'd like to start with a small physical exercise. So I'd like to ask, so we'll try to, so imagine we are all units and we are connected, we are all people, and we will engage in a mode where we'll all synchronize. So we'll all clap our hands together. Please, if you don't mind, together. Okay, exactly. So we're in a global mode. Now, try to disconnect from each other. Imagine you're isolated and try to clap asynchronously from anyone else, like in your own... Okay, very good. So this is in a way to try to explain the difference between a system that is globally integrated, that is synchronized, or segregation, local specialized processing, like each one of us is a unit that is performing its own task separately, or we're all engaged in a global mode. But both these states, although they can be relevant for natural processes, a lot of life processes in nature involve like this self-organizing patterns that involve only a subgroup of systems that engage in coordinated activity, forming what is said functional networks. So in between segregation and global integration. For example, if I would ask only the people with glasses to synchronize or only the blonde people or whatever, we would form, so it's in between. So all these three types of dynamics are important to, in a way, for the brain. It seems that the brain is using this localized, specialized processing at the local level to process auditory inputs, visual inputs, and then there's this system-level integration where things are coordinated together to form functional networks. So, in general, like this is approached as network dynamics is approached as a system of coupled dynamical systems. So in dynamical systems theory already, it's the idea is that you can represent the ensemble dynamics of homogeneous units, let's say neurons, when they are firing all together, or atoms, or stars, or things, and you treat them, and you look at their mesoscopic behavior, like the collective ensemble. And this can be described with a set of differential equations that describe how it responds over time. And then when you couple dynamical systems together, so that in the autonomous dynamical systems, then it's usually seen that the system obeys to at the macroscopic level, to self-organizing principles that are described in nature by universal laws. This is an example, for example, with migration swarms of birds and these things where you cannot understand the macroscopic dynamics by looking at an individual system alone, if you just look at one area or one neuron. So the study of network dynamics, so coupled dynamical systems, is an expanding field in physics and mathematics, but usually they use synthetic networks of either homogeneous connectivity or two communities well-defined or homogeneous delays. But in the brain, we are in the far end of complexity with non-trivial connection topologies, nonlinear couplings, transmission delays. There's heterogeneity between brain areas. So there's a way to simplify. So all these talks today have focused mostly on the structural connectivity or in the properties of the network, topological properties or graph properties of the connectivity. And indeed, this plays a major role in shaping network dynamics, but not only connectivity. So over the years, there are some papers. So the Arenas paper is quite famous because it says that synchronization reveals topological scales in complex networks. So in a way, this network that we see, we would see that the modules that are more connected together would start synchronizing together for weaker couplings, and then the whole system would synchronize. But the model they used at the time is the simple model of coupled oscillators, where in the end everything ends up synchronizing. Then, for example, Shanahan showed that you can have these metastable chimera states that is called, for example, that the blue community synchronizes, then the purple community, then the light blue, then the green. So there is this competition between different community structures that constantly synchronize. Then there are all these papers on collective dynamics in small world networks. So here the message is really, you have a structure, this structure shapes how the energy is flowing over the system over time. So behind all the concepts of graph theory that we can look at from brain networks, I'd like to show this work by Selena Tassoy, who recently joined our group in Oxford, where she shows that the connectome harmonics, which are captured by the Laplacian eigenvectors of the structural connectivity. So it's an operator, it takes into account the degree and the distance matrix, so the weights. And this system, in a way, is describing how the energy is flowing through the network. And it seems to reveal spatiotemporal patterns in the brain that are very similar to the functional networks that are found in real data. So we try to apply this to the structural connectivity. So Silene did it before at the high resolution level, at the voxel level, so with the high resolution connectome. So we tried to do it with the structural connectivity in AAL parcellation, and indeed the Laplace eigenvector, so the slowest modes seem to reveal, so there is one that is the global, everything is synchronized, is the first one, like where this mode always exists in a network. But then there's one where one network or the other, one hemisphere or the other synchronizes. And then there seem to be other patterns that are revealed in the structure. But importantly, the Laplace, so this Laplace operator, so Laplace already did this 200 years ago. So, and the beauty of it is that even with very, very sparse connectivity, so even if we just take the 10% strongest links, it still reveals the same flows. So this is what the Laplace is doing, is to capture the gradient flows through the structural connectivity. So maybe in a way linked to these cavities or kernels that exist, that are shaped by the structure, and these, in a way, kernels will reveal these modes of activity. And importantly, so what we've been finding is that if we look at the eigenvectors of the functional connectivity, they are very similar to the Laplace eigenvectors of the structural connectivity. So there seems to be a link that the functional connectivity is actually revealing this intrinsic gradient flows of energy through the connectivity matrix. But it's not a one-to-one relationship, so we need to take into account that functional connectivity is a flow, and the structural connectivity is a static network. So, looking at to model network dynamics, it's not important just to have the structural connectivity or the connection topology, but also to have a node model. So, which means each area will respond to input, so to understand how it responds, and also a coupling term, which is how the output from brain areas will translate into input to the other brain areas. So in a sketch way, it's like, so you have each brain area in the model is a neural mass, and then we will sum it by a coupling term that considers the excitatory input received from connected areas. Because it's considered that at this macroscopic scale, long-range connections via white matter tracts, fiber tracts, are mostly excitatory. And then we can consider conduction delays, because we need some nonlinearity such that the whole system doesn't fully synchronize. So we need the brain not to be always in this global synchronized state, but to be, in a way, we call it frustration in physics, and conduction delays allow to break full synchrony. And noise as well is usually used in some models. So we put weak coupling and then add noise such that the whole system never fully synchronizes. So in general, these models of network dynamics, they take into account, we can consider that each node is composed of gray matter. So there are gray matter nodes that are connected via long-range excitatory links. So each node is actually composed of a lot of neurons. And it's densely packed, it has a lot of synapses, and the idea is to model the node dynamics as a dynamical unit, a dynamical system. So for that, these networks, these densely interconnected networks, are composed of excitatory and inhibitory neurons. And it's known, for example, that the negative feedback from inhibitory neurons to excitatory neurons can drive oscillations, can have specific dynamical regimes. So this can be reduced to a set of equations that describe the mesoscopic dynamics of a single brain area. So there are, depending on the model, some consider real neurotransmitter time constants, so take into account inhibitory time scales, excitatory like NMDA and AMPA, also the ionic membrane conductances, so it takes time for ions to cross through the membrane. There are leaky ions, there are active ion channels, or some models focus directly on the oscillations that are generated by these time constants of inhibition and membrane conductance. So one of the things that is so far assumed in the models that is important to mention is that brain areas are considered for simplicity to be all identical. Because the idea was to see if you could couple identical units, so identical gray matter nodes, so it doesn't matter where they are, they are all homogeneous, but then just through the structural connectome, so the way they are connected would make them behave differently. So this is, so the heterogeneity is not at the node level, so gray matter brain areas would have homogeneous behavior, and what makes the difference in the network is how these areas are connected to each other. So this could explain why in some brain injuries or something, it's just a matter of rewiring and then you can use another part of your gray matter to perform the same function you had before. But of course, now a new model we are working on modeling because each area has specific neurotransmitter receptors, so neuromodulators. So Gustavo will talk about this on his keynote. So in general, and not to bother you too much, these models consist of sets of differential equations. So differential equations mean, so imagine the activity of a node n, so the activity x of that node varies over time. So dx dt is how the activity dxn in node n will vary over a small instant of time. And it depends on its own intrinsic dynamics plus the input received from coupled masses. So some models consider the excitatory and inhibitory pools of neurons, so they have two equations per node, so, or even more, the dynamic mean field actually considers the input currents in a node, then how these input currents are translated into a firing rate, then how these firing rates drive again the input currents. There's a simple rate fluctuation model where basically the input just increases the firing rate of a brain area or decreases its firing rate, but in a very smooth way. Or you can use more realistic models, or even really abstract models like the Hopf model or the Kuramoto model, where we are focusing more on phenomenological aspects of coupled dynamical systems in nature, like general, so we use canonical forms, and when I mean canonical form, it's or a normal form, it's an equation that describes a system, for example, that oscillates in time, so the Hopf model, it's, for example, when you have a a pendulum or something, you have a stable fixed point. And then if you add input to it, it will start kind of oscillating, and then it can return back to equilibrium. So that's the Hopf in the damped regime. So it has an intrinsic frequency, and it will return. And then you can couple these pendulums in the structural connectome and look at its dynamics and see there is an analogy, there's something coming out of this that relates to what we observe in fMRI data. So it's a phenomenological rather than a predictive model. So here again, in all these equations and the other models that can appear, the important thing is that you always have this coupling term that sums over all other nodes P. And so the activity of each node N, for the nodes that have, the models that have excitatory and inhibitory pools, you see that the coupling term is only in the excitatory term. And so this term is always scaled by this matrix CNP or Cij here, depending on the models. But you'll see after the sum symbol, there's always this CNP, and this is where the structural connectivity gets in the model. And then there is the coupling term. It can depend on basically just on the difference between their activity or on the difference between the phases of the activity. So if one is going up and the other is going down, it will take that into account. So just to show how much this is not so scary and it's not rocket science, I gave an example of how we simulate numerically this time series from these models in a very simple example. So the idea is that these systems of equations of coupled dynamical units is very hard to treat that analytically. So we can estimate fixed points, but to really see what is the dynamics coming out and the correlation matrices, it's usually important to run simulations on it. So here I just do a small random network with 10 nodes. So I define n equal 10, the total time of time steps that we will simulate. I define a random connectivity matrix with 10 nodes. So it will have here 50% connectivity. And then I initialize x, so it starts with all 0. So we have 10 nodes over 1,000 points. So we want to simulate that time series, the time series that is coming from the structural connectome. And then we, just we have to initialize the units with a given value. So I initialize them with a random value, a random phase in this case, so between 0 and 2 pi. So this is where we define x of all nodes. It's a random value. And then I define the global coupling. And for each time, after the third time, because the first two I initialize randomly, we start iteratively running, making the calculation. So at time t, so we first put it equal to the activity it had at time t minus 1. And then for each node n and each node p, the activity in node n will be its own activity plus the coupling term times the connectivity weight between nodes n and p, so the CNP term. This is where the structural connectivity matrix gets in. And here I use a term which is the difference between the phases. And I put there, so it's the value of x and t minus xp at time t minus 2. So I consider that it takes 2 time steps to travel from node n to time t. So it's homogeneous delays, but 2 time steps of delay. This is usually useful for, as I said, to break synchrony. This code is online at my page. If you just Google Joana Cabral, my codes. And I also have there the network model for the Kuramoto with time delays. So if I run that mini code that I did, and then I show here 3 different runs of exactly the same code. And we see that, so the first is the structural connectivity. It's just a binary connectivity matrix. And at each run, it generates a different structural connectivity. And every time the connectivity, the rest is all the same. So just because the structural connectivity is different, we see that the correlation patterns between the areas are different. And also the time series have different, not only So they don't just have different functional connectivity, but they also seem to be operating at different spectral scales, so at different frequencies. And this is where, for example, when Anne was talking about these cavities and these kernels that emerge in the structure, and the thing is, if you consider that there are delays between brain areas, and there are these loops naturally existing in the brain, these loops will generate oscillations. So I didn't put oscillations in the system initially, but the fact that you have, so also in our heart circuits, we have this circuitry that generates a heartbeat. So the fact that we have circuits in our brain, a lot of loops, it can be generating different spectral signatures that may be what we observe at the macroscopic level in EEG and MEG. So we observe these collective brain rhythms that could be related to these long-range loops. They may not be generated just at the local level. So after this small example, we can simulate this with real network structural connectivity, and then we can treat the data, the simulated signals. So I usually simulate like for 300 seconds, but at 0.1 millisecond resolution, so like 2 million points, and then for each area. And then we can either bandpass filter the signals, so to see if there is any spectral signature, frequency-specific connectivity, or we can use a balloon model that actually acts like a low-pass filter, so we only consider the slow fluctuations of it and see how they correlate together. So, for example, irrespective, for example, of the network model that you choose, of the node model that you choose, or the interaction term, there are some properties that arise naturally. For example, the fact that if we increase the global coupling strength, so the K, that scales all the connections equally, it scales the connectivity matrix. Obviously, if we keep increasing the coupling, we'll see more correlated activity. So if the coupling is very weak, the functional network will be more random. So the structural connectivity is the same, it's just the global coupling strength, so something related to maybe synaptic connectivity or something will make the modules or the structure more evident or less. And of course, this then has implications in graph theory. So as we change the coupling weight, which is in the x-axis, we see that the properties, the graph theoretical properties of the functional network, when it's thresholded, will change. Because of course, as the coupling is weaker, the network is more random. So we used this to the paper by Linal, also with Daniel Basset, where they studied functional networks in people with schizophrenia compared to controls. And they found all this number of graph measures that are in green, the dashed lines. So the green dashed line is the one that Linal found the graph properties for healthy participants, and the red dashed lines are the values that they found for people with schizophrenia, the FC, graph theoretical properties. And we applied the same thresholding to our simulated networks, and we found that all the changes that they reported in structural connectivity could simply be explained by a reduced coupling weight between brain areas, going in line with the idea that schizophrenia is in a way a disconnection syndrome. Okay, so to go more into detail with the models, I will explain here one of the most reductionist approaches, so the one we used for that model and for others, is where we consider each brain area as an oscillator. Because it is reported that neural masses with excitatory and inhibitory neurons can engage in gamma frequency oscillations, which is detected at the LFP, local field potential. And so the idea here is to assume, okay, let's see what happens when you just couple oscillators together in the structural connectome. Then now we are also considering the amplitude dynamics using Hopf, but this was to make the link with a lot of theoretical papers from theoretical physics who study the dynamics of coupled oscillators. So this was the model was initially described by Kuramoto in 1984, and it describes beautifully a lot of systems of coupled oscillators in nature. But one of the things is that coupled oscillators in nature usually couple to their nearest neighbor. So there isn't a time delay between areas. And our brain has this specific structural topology that has this physical distance between brain areas that makes some connections to be longer than others. So the idea is to introduce time delays in the Kuramoto model, which has been done already. There are some studies, not many, but some studies studying the dynamics of the Kuramoto with time delays, with homogeneous time delays, with heterogeneous time delays, and it really shows that it generates metastability, generates collective frequencies, and generates many systems, many dynamical regimes. So this model, basically, the phase of each brain area depends on its own natural frequency. So here we consider the gamma range, so 40 hertz, all identical oscillators at 40 hertz, like all coupled pendulums. And then we have this coupling term, where this is the global coupling. Like I said, weak coupling, everything is segregated. High coupling, everything is synchronized. We need an intermediate coupling. Then this is the structural connectivity, the real. And this is the conduction delay that we take as a matter of the distance between brain areas, or the fiber length, depending on whether we have that available or not. And then we show that for weak coupling, we have incoherence. Then for high coupling, everything is synchronized. And for intermediate coupling, we find a regime of metastable cluster synchronization. So where we find some networks synchronized, then others synchronize. So the order parameter, so what is plotted in the color over time, the R, is the order parameter of each cluster of brain areas over time. And then below are snapshots of the dynamics. But the middle one, you would see that different brain areas, so here the blue and the red seem to be more synchronized, but over time, this would be switching. Okay, so just with this simple model, so the idea this was that it was pretty phenomenological and less complex than other more realistic neural mass models. The idea was to show that only using the structural connectivity, so the first column shows the structural connectivity of the left posterior cingulate, the connectivity with the rest of with all the other brain areas. So in red, it's the posterior cingulate, and we see how much it connects structurally with the other brain areas. And then the second column is how the bold is correlated in real data, average across 10 subjects. And then in the third column is how we model these correlations between brain areas. So we can indeed see that this metastable synchrony could be explaining some of the phenomena we are observing. But, there are many models, and many models are indeed showing a capacity to fit this static bold functional connectivity matrix. But we found it's important not only to fit those correlation patterns, but also to see how much these correlation patterns evolve over time. So if indeed you have different functional networks that synchronize over time, and also see if you can explain the frequency specificity of functional connectivity that is detected with MEG. So what does this mean? It means that in MEG, they found that some functional networks are more connected in a specific frequency range. So it would be like if some of you would like to communicate with each other, like a subgroup, like everyone with glasses, you would clap in a different frequency from the others, but you would synchronize only at that frequency. And this is usually found around the alpha and the beta ranges in EEG. So they found simultaneous power fluctuations on these oscillations, which means that those units increase together in this oscillatory regime. So we used also the Kuramoto model. So we used real MEG data from 25 subjects. And so we filtered the MEG data, source projected into the 90 AL areas, then took the envelopes in each frequency band, see how much they correlate over time. So we have a FC profile that is frequency specific. And then we did the same with the Kuramoto. So I recall that each brain area is oscillating only at 40 hertz, but then there are realistic time delays. And those time delays actually drive, so we found that, so in the range where we have our oscillations, the last two columns, it's the real MEG and the model. We see that the connectivity is only with the most connected areas. And then in the range of alpha and beta, so between 10 and 21 hertz, we find that it's where we have more connectivity. So the brain areas seem to communicate in that frequency range, both in real data and in the model, and also at lower. So we also find this structure also in alpha, but it's less, so it's a slightly different functional network that is revealed in alpha than in beta. But still, mechanistically, this is interesting. So again, going back to the structural connectome, so this is, we find that there are two variables here in this model that were important, which is not only the coupling weight, so this is each plot is a parameter space where the vertical axis is the coupling weight and horizontal axis is the time delay. And we find that the optimal fit to MEG data is found for a certain value of coupling, so not very strong and not very weak, but also a certain value of delays. So you need at least 10 milliseconds mean delays between brain areas, which is in the range of biologically plausible transmission speed. And we show here, well, it's a bit complex, but anyway, there's an important range of metastability that must be generated through these delays that allows, so it's in the last column, we see this red range where it shows that we observe the emergence of alpha and beta band fluctuations in the last column, and whenever we disrupt the connectome by randomizing the connectivity or putting homogeneous couplings or homogeneous distances, we break this system, so this emergence of collective frequencies that reveals specific spatial patterns. So overall, well, for example, this mechanism, in theory, it could work then even with more realistic models if you put their time delays. But the thing is that more realistic models then are harder to make the link with theoretical works. So collective frequencies with time delays have been described in the literature with simple homogeneous networks. And even if they emerge in more complex networks, then it's really hard to understand if it comes from the inhibitory time constant or from the membrane conductance time constant. So in a way, there are different degrees of biophysical realism. So they are sorted here. I tried to do for this review, in collaboration with the reviewers and everything, a table that would compare the different models, and we sort them vertically according to their biophysical realism and how much they explain. So the idea here was to see especially what is the mechanism, right? Rather than finding what is the optimal fit that they achieve with the data, so it doesn't matter if it's 0.4 or 0.5 or 0.7, because this depends a lot on the parcellation you use and the data you have, but more like, do you generate slowable signal fluctuations? Do you achieve a state where the system doesn't fully synchronize, but you observe like metastable cluster synchrony or self-organizing patterns as are described in more realistic models? Also, does this change over time, and does this have specific frequencies? Do these cluster synchrony also lead to the emergence of collective frequencies in the system? So all of these things, that's what I try to put here together in this table, are important and are part of this large collaborative project that has been undergone over the last 10 years, where we're trying to link brain dynamics, not just functional connectivity, but the whole spatiotemporal properties of brain dynamics at the macroscopic level to the structural connectome. See what is emerging from it. And, well, I like this sentence saying, all models are wrong, but some are useful. So in general, I just want to say that whole brain network models are useful tools to investigate these self-organizing principles governing the formation of functional networks. In a way, as I see this, the functional networks are attractor states that are shaped by the brain's network structure. And these networks, these attractor states, could be driven by kernels, could be driven, these connectome harmonic modes. So this flows through loops that generate the more stability of certain patterns rather than others. So it's not like a totally incoherent system. Some patterns seem to be more stable than others. And when we are in the critical border of this multi-stability of different attractor states, configurations that our functional connectome can explore, when we are at rest, we would be exploring this landscape of attractors, and whenever we would start a task or do something, we would stabilize a specific network. That's it. Yeah, I'd like to thank exactly on time. So my collaborators, Martin, Gustavo, and now Nuno Souza in Portugal, they've all been really supportive. And yeah, that's it.